Before the early 1990s, a 25-year-old diagnosed with HIV had a 50% chance of being dead by age 30 and a 0% chance of living to 55. But today, HIV is considered by many to be manageable. Dr. David Ho discovered that when 3 different drugs are taken as a cocktail, drug resistance is deferred. Since then, HIV drugs have saved 3 million years of life in the United States. Now, HIV has become another chronic condition, like thyroid imbalance, diabetes, or heart disease. And, thanks to pharmaceutical progress, HIV is much easier to manage. Newer drugs combine 2 or more agents. They can be taken on longer intervals, with side effects that are less toxic and easier to live with. While effective cocktails are expensive, for many people insurance covers the cost, so even a therapy that costs $25,000 to $30,000 a year is manageable. After all, chronic conditions without cures or durable therapies are expensive. With all these advances in the last decade, some have declared mission accomplished. But is that what we should settle for? Are we really done with HIV? What if we could completely change the management of HIV? Today, experts estimate that 10 to 15% of people newly infected with HIV have strains of the virus that are resistant to all existing drugs. A decade ago, this statistic was closer to 1 to 2%. HIV is on the march. Can this march be stopped, or will resistant strains emerge for which no therapeutic regimen can be found? Current HIV therapies work by suppressing the HIV virus, interfering with viral replication, blocking the virus from infecting new cells. But either way, existing mechanisms put pressure on the virus. The virus strains that can evade this pressure are the ones that survive. It is clearly time for a new treatment for HIV that can work in concert with existing therapies, while providing a real possibility for a durable cure. At Koronis Pharmaceuticals, we are pioneering a new way of fighting HIV. Viral decay acceleration. No suppression, inhibition, or blocking, but instead turning the virus's natural defense mechanism, rapid mutation, into a weapon against the infection. Koronis has found a way to accelerate viral mutations that drive the virus to a point where it is incapable of further viability. Thus it causes the virus to become extinct. And extinction means a durable and lasting therapeutic benefit. Viral decay acceleration represents a novel approach that could lead to a cure. Here is how it works. Viral decay acceleration causes extra mutations to form in the HIV genome during replication. These extra mutations get inserted randomly across the HIV genome. The random location of an extra mutation does not put pressure on the virus. No pressure means no resistance. In vitro studies have shown that viral decay acceleration works in a 2-stage process. Stage 1 is the crippling phase, in which extra mutations accumulate, leading to decreased viability of the virus, until a tipping point is reached, or Stage 2, the collapsing phase, in which no viable HIV virus remains. Conversely, traditional antiretrovirals produce a rapid decrease in viral load, but over time, resistant strains emerge and viral load increases. Over the last 10 years, the development of viral decay acceleration at Koronis has been highly focused and methodical. In 3 long-term in vitro experiments, viral decay acceleration caused the permanent eradication of HIV. Viral decay acceleration has been shown to be safe and well tolerated by over 80 patients in early human trials. There has been no evidence of drug resistance. Results from the early human clinical trials have shown conclusive evidence that viral decay acceleration causes a significantly increased rate of mutation formation in HIV. Koronis has a strong intellectual property estate. It covers the viral decay acceleration mechanism, a composition of matter patent on the lead HIV compound, and for viral decay acceleration compounds against hepatitis C. Where does viral decay acceleration go from here? Koronis needs a sophisticated development partner to complete late-stage development that allows for a full-scale Phase 2 study to determine how long a patient must take the drug before a clinically meaningful reduction in HIV levels occurs. If successful, One or more Phase 3 trials will be necessary to gain regulatory approval for the drug. Patients could then begin to use a drug that could change the way HIV is treated. To learn more about viral decay acceleration and a new approach to treat and perhaps cure HIV, contact us.